back to this session on transcription. And the learning goals are 1. Briefly highlight the 2 steps of gene expression. 2. Explain the role of RNA polymerase in transcription. 3. Examine why both the strands of DNA are not used as templates during transcription. 4. Identify differences in the structural genes in prokaryotes and eukaryotes. 5. Describe the 3 steps of transcription with illustration. And 6. Outline the steps of RNA processing in eukaryotes. DNA, which is our genetic material, carries the instructions of life known as genes in its long nucleotide sequence for producing either proteins or RNA molecules. And the process of producing functional proteins or functional RNA is called as gene expression. Gene expression process is mostly completed in 2 stages. The first step is transcription. This involves the copying of information in a particular segment of DNA into an mRNA. The second step is translation, in which information in the mRNA, which is coded in a chain of nucleotides, is decoded into a chain of amino acids or protein. Taken together, transcription and translation constitutes the 2 events of the central dogma of molecular biology, which states that the flow of genetic information is DNA to RNA to protein. In this video, we are going to be learning about the first step of gene expression, which is transcription. Unlike the process of DNA replication, where the entire sequence of the DNA is copied to make daughter DNAs, during transcription, only a segment of DNA is copied into RNA by an enzyme known as RNA polymerase. The chief role of RNA polymerase is to synthesize a new RNA strand from a DNA template. And the basic principle followed to synthesize the new RNA strand is very similar to that of DNA replication, where RNA polymerase synthesizes a new RNA strand by following the rules of complementary base pairing, in which for each T on the DNA template strand, an A is added in the RNA strand. For each C, a G is added. For each G, a C is added. However, for each A that is encountered on the DNA template strand, RNA polymerase is going to be adding a U in the RNA strand. This is because thymine is replaced by uracil in RNA. And another striking ability of RNA polymerase is that it can initiate the synthesis of RNA strand without primer. And just like the DNA polymerase, RNA polymerase can always be synthesizing the new RNA strand in the 5' to 3' direction, which is to say that the enzyme can only be adding free RNA nucleotides, which are also known as RNTPs, at the 3' end of the growing strand, and hence the new RNA strand synthesis will proceed in 5' to 3' direction. Eukaryotic cells have 3 main types of RNA polymerases, which are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. These 3 RNA polymerases differ mainly in terms of the type of RNA that they transcribe. RNA polymerase 1 is located in the nucleolus, and this enzyme is responsible for the transcription of rRNA, R standing for ribosomal RNA. The RNA polymerase 2, which is present in the nucleoplasm, is involved in the transcription of mRNA, M standing for messenger RNA. While the third type, known as RNA polymerase 3, again present in the nucleoplasm, is responsible for the transcription of tRNA, where T stands for transfer RNA. So these are the 3 types of RNA transcribed by 3 different types of RNA polymerases in eukaryotes. In prokaryotes, however, they have only one type of RNA polymerase which can transcribe all the 3 types of RNA. Another aspect of transcription that differs from DNA replication is the use of template strand during transcription. Unlike DNA replication, where both of the strands of the DNA are used as templates for the synthesis of new DNA strand, in transcription, out of the two strands of the DNA double helix, only one of the strands of the DNA is used during transcription. And this is the strand which is oriented in 3' to 5' direction. This 3' to 5' directed strand, since it is used for the synthesis of new RNA strand, it is named as template strand, or it's also called as antisense strand. The other strand of the DNA, which is oriented in 5' to 3' direction and which is not used during transcription, is called as the coding strand, or it's also called as sense strand. You might wonder why the non-template strand is given the name coding strand or sense strand. This is because if we look into the sequence of nucleotides in the non-template strand and compare it with the sequence of nucleotides in the newly synthesized mRNA strand, we find that the sequence of nucleotides in the non-template strand or the coding strand is completely identical to the sequence of nucleotide in the newly synthesized mRNA strand. This makes sense because RNA polymerase synthesizes the new RNA strand by following the complementary base pairing rule. So this results in a complementary mRNA whose sequence is completely identical to the sequence of the non-template strand or the coding strand, except for uracil in place of thymine and ribose sugar in mRNA. Well, you may think why both the strands of the DNA are not used as templates during transcription. To understand this, let me first draw your attention to the two red strands here, which denotes the two strands of a DNA segment. We know that the two strands of the DNA are complementary to each other but not identical. And because of such a scenario, if we are to be using both of the strands of the DNA as templates during transcription, it would then be resulting in the synthesis of two different mRNAs. So the two blue strands here are indicating the newly synthesized mRNA. Now, the sequence of nucleotide in the mRNA, which is synthesized by using 3' to 5' template strand, reads as AUG-GCG-AAA in the 5' to 3' direction. Whereas, the sequence of nucleotide in the newly synthesized mRNA using the 5' to 3' DNA template strand reads as UUUCGCCAU in the 5' to 3' direction. So you've noticed that the sequence are entirely different in the two mRNAs. And not only that, if these two different mRNAs are to be proceeding into the translation, then they are going to be producing two different polypeptides. Because since the sequence of the nucleotides are different in the mRNA, it would result in the different sequence of amino acid in the polypeptides. Such a synthesis of two different polypeptides from the same region of the DNA at a time would only be complicating the process. Another problem is the two mRNA strands being complementary to each other. So they can pair up and form double-stranded RNA. And these double-stranded RNA cannot be translated into proteins and are only to be degraded, making the whole process wasteful. So these are some of the reasons why both the strands of the DNA are never used as templates during transcription. It's only the 3' to 5' strand which is used as template strand for the synthesis of mRNA or for the synthesis of RNA. As I said earlier, during transcription, a segment or stretch of DNA is transcribed into an RNA. And such a segment of DNA is referred to as transcription unit. Transcription unit essentially carries the gene that needs to be expressed. In each transcription unit, there are three components, which are a promoter followed by the structural gene and a terminator. The promoter is a DNA sequence that specifies where transcription should begin. And the structural gene is the actual DNA sequence that carries the coding sequence or the genetic code, the information for the synthesis of polypeptide or protein. And the terminator is a DNA sequence that specifies the end of transcription. Basically, promoter is in front of the structural gene and terminator is at the end of the structural gene. The structural gene is mostly polycistronic in prokaryotes, which means that the structural gene in a transcription unit contains more than one gene in prokaryotes. On the other hand, structural gene in eukaryotes is mostly monocistronic, which means the structural gene in a transcription unit contains only one gene. At the same time, the structural gene in prokaryotes is said to be uninterrupted gene, which means it contains continuous coding sequences for polypeptides. The structural gene in eukaryotes is split gene or interrupted gene, consisting of stretches of coding sequences called as exons, interrupted by stretches of non-coding sequences called as introns. So exons are coding for polypeptide, while introns are not coding for polypeptides. And in our eukaryotic DNA, the gene is said to be a split gene because we carry stretches of exons interrupted by stretches of introns. Next, we will briefly go over the steps of transcription. On the whole, transcription in all organisms occur in 3 steps. The first step is initiation, during which an initiation complex is formed at the promoter region of the DNA by the assembly of DNA, RNA polymerase and several protein factors. You know already that during transcription, a segment of the DNA called as a transcription unit is copied into an RNA. In my illustrations over here, the strand of the DNA which is drawn in red color is the 3' to 5' template strand. And here is the promoter sequence of the transcription unit followed by the structural gene and ending with a terminator sequence. So during initiation, RNA polymerase binds to the promoter sequence of the transcription unit. And binding of the RNA polymerase to the promoter sequence is mediated by a sigma factor in prokaryotes, while binding of the RNA polymerase to the promoter sequence is mediated by several transcription factors in eukaryotes. And after binding to the promoter sequence, RNA polymerase then opens the double helix or unwinds the double helix and forms a structure called as transcription bubble. And in a transcription bubble, we can have somewhere about 10 to 20 exposed bases at a time. And RNA polymerase then initiates the synthesis of the first few RNA nucleotides. So it then synthesizes the first few RNA nucleotides by reading the template in the 3' to 5' direction from the starting point. Initiation of the new RNA strand synthesis from the start point is followed by the next step, which is elongation. The basic mechanism of elongation remains conserved or remains same in both prokaryotes and eukaryotes. During elongation, RNA polymerase moves along the template strand in the 3' to 5' direction and continues copying the information in the template strand by adding free complementary RNA nucleotides called as RNTPs one by one at the 3' end of the growing strand. You know, prokaryotes lack a membrane-bound nucleus and their free RNTPs are all suspended in the cytoplasm of these cells. In fact, transcription itself is taking place in the cytoplasm in prokaryotes. In the eukaryotes, however, we have a distinct membrane-bound nucleus and our transcription occurs inside the nucleus. So, the free RNTPs in the eukaryotes are present in the nucleoplasm. Since RNA polymerase adds complementary RNTPs at the 3' end of the growing strand, synthesis of the new RNA strand always proceeds in the 5' to 3' direction. Even though the basic mechanism of elongation remains same in both prokaryotes and eukaryotes, you might be able to observe a difference in the RNA transcript in the prokaryotes and eukaryotes. The RNA transcript that is produced in the prokaryote is called as a mature mRNA or an uninterrupted transcript. The RNA transcript obtained in the eukaryotes is called as a pre-mRNA or an interrupted transcript. The difference in the prokaryotic and the eukaryotic RNA transcript can directly be associated with the difference in the structural genes in prokaryotes and eukaryotes. If you can recall, we learned that the structural gene in prokaryote is an uninterrupted gene, while the structural gene in eukaryote is a split gene or an interrupted gene. So the uninterrupted structural gene in prokaryotes resulted in the production of an uninterrupted transcript or a mature mRNA, whereas the interrupted or split gene in eukaryotes have resulted in the production of an interrupted transcript or a pre-mRNA. The third and the last step of transcription is termination. During termination, RNA polymerase stops transcription of RNA from the DNA template strand. In prokaryotes, termination can be mediated by a protein called as Rho factor, which binds to the RNA transcript. And binding of the Rho factor destabilizes the entire complex and causes the release of RNA transcript and the dissociation of RNA polymerase from the DNA template. Contrastingly, in eukaryotes, termination of the transcription occurs several sequences beyond the terminator region. And the exact mechanism of termination in eukaryotes is still not clear. Anyways, the RNA transcript produced in prokaryote is a mature mRNA containing continuous coding sequences. For this reason, mature mRNA in prokaryote is ready for translation right away. The primary transcript in eukaryote is called as a pre-mRNA. This is because pre-mRNA is consisting of stretches of coding sequences called exons interrupted by stretches of non-coding sequences called as introns. And introns, since they are not coding for polypeptide, are to be removed from the pre-mRNA. So, pre-mRNA or the primary transcript in eukaryotes needs to undergo an additional processing before it is exported to the cytoplasm for translation. As you can see, the primary transcript in eukaryote is an interrupted transcript consisting of exon sequences interrupted by stretches of non-coding intron sequences. Such a pre-mRNA or primary transcript in eukaryote undergoes through an additional processing step collectively known as RNA processing. Through the sequence of events during RNA processing, a mature mRNA is generated from a pre-mRNA in eukaryotes. And RNA processing basically involves three steps, which are 5' capping, 3' tailing, and splicing. During 5' capping, methylguanosine triphosphate is added at the 5' end of the pre-mRNA. And in the 3' tailing step, which can also be called as polyadenylation, we have the addition of poly-A nucleotides. Hundreds of adenine nucleotides are added to the 3' end of the pre-mRNA. And the most important step of RNA processing is the splicing event. During splicing, we have the removal of non-coding intron sequences by complexes called as spliceosomes. And after the removal of introns by the spliceosome, we have the subsequent rejoining of exons into a continuous strand. And this results in the generation of a mature mRNA. Such a mature mRNA consisting of only exon sequences is now ready to be exported out of the nucleus into the cytoplasm for translation. That's it on transcription and RNA processing. See you back with my next video on genetic code.